Nick translation is a crucial technique in molecular biology used for labeling DNA with radioactive or non-radioactive markers. Let's explore how it works and its applications. Nick translation involves two main enzymes. DNase I introduces single-strand breaks, or NICs, in the DNA. DNA polymerase I then removes nucleotides around the Nick and replaces them with labeled nucleotides. This process results in a DNA strand labeled with detectable markers at multiple points. Nick translation is commonly used for generating labeled DNA probes for hybridization experiments like southern and northern blotting. These probes can be radioactive or non-radioactive, depending on the application. In fish, Nick-translated probes labeled with fluorescent dyes are used to map chromosomes and detect gene amplifications or deletions, especially in cancer research. Nick-translated probes hybridize to specific DNA or RNA sequences, allowing detection and quantification of target nucleic acids in complex samples. CGH uses Nick-translated probes to compare genomic content between samples, identifying chromosomal alterations such as gains or losses in cancer studies. Nick translation generates probes for screening libraries or mapping genes, aiding in the identification and isolation of specific DNA fragments. Nick-translated DNA is used in DNA footprinting to study protein-DNA interactions, helping to identify specific binding sites of DNA-binding proteins. Nick translation is a versatile and essential technique for DNA labeling in molecular biology, with applications ranging from probe generation to genomic analysis. It remains a valuable tool for researchers.